All right, today we're diving deep into the liver. And I don't just mean as some organ in your body, but as a total masterpiece of biological engineering. We're going to take a microscopic journey, peeling back the layers to see how its incredible structure is perfectly built to function as your body's master biochemical factory. So let's get into it.
So here's the game plan for our journey. We'll start big, looking at the whole organ. Then we'll zoom right into its core functional unit, this thing called the lobule. We're going to follow the unique path blood takes through it, pinpoint the exact spot where all the magic happens, meet the specialized cells doing the actual work, and then, at the end, we'll connect all the dots to see how this brilliant design serves its purpose.
All right, let's kick things off with Section 1, Anatomy and Function. Before we go microscopic, we've got to get our bearings. Let's figure out where the liver actually sits in the body and what its main jobs are on the grand scale.
Okay, so the liver is a beast. It's our largest internal organ, tucked up under your diaphragm in the upper right part of your belly. But honestly, its size isn't even the most interesting part. Where it gets really cool is how it's plugged into your body's circulatory system.
And this right here is where the liver's design is just so incredibly clever. It has a dual blood supply. Now sure, like other organs, it gets fresh oxygen-rich blood from the hepatic artery. But, and this is the game changer, it also gets a massive flow of blood directly from your intestines through the hepatic portal vein. This blood is packed with all the nutrients you just absorbed. This is a huge deal. It means the liver gets first dibs, the first chance to process, store, or detoxify everything you've just eaten.
So, with this VIP access to your blood, what's it actually doing? Well, it's a non-stop factory, really. It's cranking out bile to help you digest fats. It's detoxifying things like drugs and alcohol. It's making cholesterol. It's balancing your blood sugar. It even takes waste products and turns them into urea. To pull off this incredible list of jobs, it needs an equally impressive internal structure.
Okay, so that's the big picture. Now, let's grab our virtual microscope and zoom way in. Because to really understand how the liver does all that stuff, we need to look at its fundamental building block, the hepatic lobule. So your entire liver, it's basically made up of millions of these tiny, repeating hexagonal units called hepatic lobules. You can think of them as the liver's individual computer chips or processing centers, each one a self-contained, perfectly organized little factory. And wow, check out this micrograph. It shows that classic hexagonal shape perfectly. See that dot right in the middle? That's the central vein. That's the drain. And then, at each of the 6 corners, you find what's called a portal area. Those are the supply lines. This incredibly simple layout, supply lines on the outside, a drain in the middle, is the absolute key to how the whole thing works.
So let's follow the blood. Let's trace its path as it flows through one of these tiny lobules. And I can't stress this enough. The direction of that flow, from the outside in, is everything.
It's really a beautiful 4-step journey. First, blood comes in at the corners from those 2 different sources. Then, that blood mixes together and starts to flow through these special channels called sinusoids, heading inward toward the middle. Third, it all gets collected by that central vein, the drain. And finally, that vein carries it out of the liver. A simple, elegant trip from the periphery to the center. This diagram nails it. See those arrows? Blood from both the portal vein and the hepatic artery pours in at the corners. Then it slowly percolates through those sinusoids, flowing past all the liver cells and making a beeline for that central vein. This slow, deliberate flow is by design. It's meant to maximize the amount of time the blood spends in contact with the cells.
All right, so we've got the direction of flow down, but let's zoom in even closer, right down to the nitty-gritty, to the actual interface where the blood and the liver cells meet and do business. This is where it all goes down.
Now, these channels, the sinusoids, they're not your average capillaries. For one, they're much wider, which really slows down the blood flow. And critically, their lining, the endothelium, is fenestrated, which is just a fancy way of saying it's full of tiny holes, like a strainer. And these leaky channels run right between plates of the main liver cells, the hepatocytes.
So, that begs the question, right? If the blood is in the pipe and the cell is next to the pipe, how do the goods actually get exchanged? There has to be some kind of go-between space for that transaction to happen.
And the answer is the space of Desai. It's this incredibly tiny fluid-filled gap that sits between the leaky sinusoid lining and the liver cell. Blood plasma literally seeps through the holes in the sinusoid and fills up this space, bathing the liver cell directly. And to make it even more efficient,
The cell surface facing this space is covered in microvilli, tiny fingers that just massively increase the surface area for absorption. Think of it as the ultimate loading dock. This whole setup leads to a really powerful idea, which the source material puts perfectly. At least 2 surfaces of a hepatocyte is surrounded by blood. The entire structure, every plate and every channel, is engineered to maximize this contact, to make sure the exchange is constant and incredibly efficient.
Okay, we've got the architecture down. Now it's time to meet the crew, the specialized cells that actually live and work inside this amazing structure.
So you've got 2 main characters here. First are the hepatocytes. These are the superstars, the workhorses doing all the heavy metabolic lifting. They're the polygonal cells that form those plates we talked about. Then you have the Kupffer cells. Think of them as the bouncers or the security team. They're actually macrophages that just hang out inside the sinusoids, munching on old red blood cells and any bacteria that might have slipped through from the gut. But wait, there are more. Let's meet 2 other specialists. Over here, hiding out in the space of Dece, we find stellate cells. Normally they're pretty chill, they just store vitamin A. But if the liver gets injured, they can get activated and start producing scar tissue, which leads to fibrosis. And then, on the other side, you've got the cholangiocytes. These are the simple cuboidal cells that line all the little bile ducts, and their job is to transport the bile that the hepatocytes produce.
And we can't forget the endothelial cells. We've mentioned them, but they are so important. They are the gatekeepers. They're the ones that form that super thin, porous, fenestrated lining of the sinusoids. Without their unique, sieve-like structure, that critical exchange happening in the space of Dece just wouldn't be possible.
All right, this is the grand finale. For our final section, let's tie everything we've seen together. We're going to see exactly how this stunning microscopic architecture is perfectly, and I mean perfectly, engineered for the liver's job. So let's put it all together with one big question. What makes this whole design so ridiculously perfect for a job like detoxification?
Well, it's because every single piece of this puzzle fits perfectly. You've got the dual blood supply bringing toxins straight from your gut to the liver's front door. You've got the wide sinusoids that slow the blood to a crawl, giving the cells plenty of time to work. The fenestrated endothelium in the space of Dece create a direct, open highway for those toxins to get from the blood to the cells. And finally, the hepatocytes themselves have a huge surface area, ready to process whatever comes their way. It's a perfect system.
So you see, when you look at it this way, the liver isn't just another organ. It is a jaw-droppingly complex biochemical factory, where every single microscopic detail is designed with a purpose. It really is a masterpiece of biological engineering.